Okay, guys, we are going to pick up with Chapter 6, Video B. We left off right before the 3 stages of cell respiration. So again, if you'll make sure you have your objectives in front of you as well as your Chapter 6 notes, we are going to start with objective number 6, list the 3 stages of cell respiration, describe the location and energy yield of each stage. So there are 3 main stages of cell respiration. Now, this is just an overview. So step 1 is called glycolysis. It occurs in the cytosol. So it's going to begin cell respiration. It's going to break down glucose into 2 molecules of a compound called pyruvate. Now, glucose is 6 carbons. Pyruvate is a 3 carbon. So basically, we take glucose and we break it in half, okay? We end up producing 2 ATP at the end of glycolysis. Now, stage 2 is oxidation and the citric acid cycle. Some people call this the Krebs cycle, but we mainly use citric acid. Now, this is going to actually take place in the mitochondria. So we have actually moved into the mitochondria on stage 2. It's the complete breakdown of glucose to carbon dioxide, and we're going to produce 2 ATP, which is going to supply the last stage of respiration with electrons. So honestly, y'all, stage 1 and stage 2, we get 2 ATP from each. They're really just to drive the 3rd stage. They're to provide those electrons and energy for allow the 3rd stage. Because the 3rd stage is where we get most of our ATP from. But glycolysis starts in the cytosol, so we're right outside the mitochondria, and then we move into the mitochondria for the citric acid cycle. All right, so right here, stage 1 glycolysis, we take glucose, split it into pyruvate. We end up with some ATP. Then pyruvate moves in and is oxidized, and it enters into the citric acid cycle. Okay, so it's going to release a little bit of CO2. Citric acid cycle releases some CO2 and ATP. Citric acid also releases NADH and FADH, but again, we'll discuss that when we get into the details. And then step 3 is oxidative phosphorylation, which is the electron transport chain and chemiosmosis. This is where we produce the water, we use oxygen, produce water, and we get a whole bunch of ATP, okay, from those. Again, we're in the mitochondria. So this is just a little close-up, a better image. This is in your book on page 117, all right? Really, really great images there. Now, that gets us to step 3, which is oxidative phosphorylation. It involves electron transport chain and chemiosmosis. So what happens is our electron carriers, NADH and FADH, they're going to bring electrons to the ETC, which is electron transport chain, and we are embedded within the inner mitochondrial membrane. All right, so that's where we're at, inside mitochondrial membrane. Most of the ATP, as I told you a minute ago, most of the ATP made in cell respiration is generated at this stage. The electrons are passed down the electron transport chain. They finally come to oxygen, which is reduced. That means it gains a hydrogen electron, and of course we end up with water at the very end. So of the 3 main stages of respiration, which one does not take place in the mitochondria? And you should know, of course, that would be glycolysis. It's the only one that's not taking place in the mitochondria. All right, so now we are going to go to objective number 7, describe glycolysis and define substrate level phosphorylation. So glycolysis is going to harvest chemical energy by oxidizing glucose and turning it into pyruvate. All right, remember, oxidizing means we are going to release electrons. All right, so we're going to split it up. So ATP is used to prime glucose. The glucose is split into 2. There are 3 carbon, we call intermediates, that are oxidized to produce 2 pyruvates. We end up with 2 ATP and 2 NADPH. That NADPH needs to be fixed. I'll fix that in your notes. That looks kind of strange. But anyway, we end up with 2 ATPs and 2 NADHs. Now, ATP is actually formed by substrate level phosphorylation. What this means is we take a phosphate group and we transfer it to ADP. ADP is adenosine diphosphate, and it only has 2 phosphate groups. So if we add a third phosphate group to it, we end up with ATP. And this is just, again, going to happen in glycolysis. We're in the cytosol. Remember, we're in the cytosol. That's the location. And it's called substrate level phosphorylation. Again, we only get 2 ATP. And then we also end up with 2 pyruvates and 2 NADPHs. But that's going to help drive our next process. Okay, so right here, in glycolysis, we're outside. Remember, look, we're not in the mitochondria. We're over here in the cytosol. We start out with glucose. We are going 2 ADPs and 2 Ps. If we put these 2 Ps onto those 2 ADPs, we get 2 ATPs. NAD, 2 NADs, you're going to pick up 2 hydrogens. They're going to become 2 NADHs. And then in that process, we split glucose in half, and we end up with 2 pyruvates, 2 ATPs, and 2 NADHs. All right, so that's what we end up with at the end of glycolysis. All right, and that's just a closer picture, same thing that we just looked at. And this is in your textbook on page 118. Now, in this, the enzyme, all right, here's our substrate, phosphate, our ADP. There's an enzyme that is going to add that phosphate to this ADP to give us ATP, all right? And notice there's an enzyme that's got a special shape, that active site, snuggles in that phosphate and ADP, and it's going to put these things together, and it's going to release the product. And remember, the enzyme's not changed, all right? That's a review from the last chapter, okay? So for each glucose molecule process, what are the net molecular products of glycolysis, okay? So if you look at that question, I'll give you a chance to kind of think about an answer. You can pause the video right here if you need to. But for each glucose molecule process, what are the net products? So the net products would be 2 pyruvates, 2 ATPs, and 2 NADHs. All right, so I hope you got that answer right, because those are the products at the end of glycolysis, okay? Now, there's multiple reactions in glycolysis that help split glucose. Figure 6.8 and 119 gives you the details of that. It shows kind of simplified structures, and there's nine, yes, you read that right, 9 reactions of glycolysis. Lucky for you, you don't have to know all of those, but I put it in here just because it's an example of metabolic pathways and how each chemical step feeds into the next. So, in other words, the product of one reaction is going to be the reactant for the next reaction, and then that product is the reactant for the next one. So the compounds that form between initial reactants and final products are called the intermediates. So if you've got 9 chemical reactions, you have a whole bunch of intermediates before you get to your overall final product, okay? So the energy investment actually consumes energy. In this phase, there's 2 ATPs that have to be invested, and then they're split into 2 small sugars. So the first 4 steps actually use energy, and the last 5 are going to actually yield or produce that energy, okay? So, again, you don't have to memorize this. This is non-objective. I just think it's interesting that you know all these steps that have to go into what actually happens, okay? All right, now we are moving in our objective to number 8, describe the citric acid or Krebs cycle, state the number of cycles required to break down 1 molecule glucose, and define oxidative phosphorylation. So this is step 2, the citric acid. It completes the energy-yielding oxidation of glucose. So we're going to completely break down glucose here. And that oxidation of pyruvate, because remember we ended up with pyruvate at the end of step 1. So pyruvate is going to move in the mitochondria, and it's going to be turned into something called acetyl-CoA. We're also going to release some carbon dioxide and NADH. Okay, now for each turn of the citric acid cycle, we get 2 carbons from acetyl-CoA, 2 carbon dioxides, 3 NADHs, and 1 FADH2. Now, I want you to be really careful here. In order to completely break down a glucose, we have to turn the citric acid cycle 2 times, okay? That's because we've got 2 pyruvates, okay? Remember, 2 pyruvates are going to come in here. 1 pyruvate is going to form 2 acetyl-CoAs, 2 carbon dioxides, 3 NADHs, and 1 FADH2. So double all those numbers, and that's going to give you the total amount that's produced at the end of citric acid. All right, so we're going to have 4 carbon dioxide, 6 NADHs, and 2 FADH2s released. And again, we still only get our 2 ATP, but that's not really the point here. We have totally broken down glucose, and we've completely, turned it into CO2. So, and then we also have the acetyl-CoA. But that's important, for you guys to understand, is that this has to turn 2 times to completely break down glucose, because we got 2 pyruvate. So 1 pyruvate goes, and then the second pyruvate goes. And you can see we end up with this acetyl-CoA, the 2 NADHs, our FADHs, as well as our ATPs. All right, so here's the citric acid that goes around twice. We are going to produce 2 carbon dioxides, 3 NADH, ATP, and then FADHs, double those numbers to get the total amount for glucose. So right here, for 1 pyruvate, again, we bring that pyruvate in, we're going to form CO2, acetyl-CoA, NADHs, and then we also are going to produce FADH2 when we get to the very end of citric acid, okay? Now, there is an image in your book, just like we had for glycolysis. This is a multiple-step process, so there are multiple chemical reactions that have to finish dismantling glucose. There are 6 major steps, and it's figure 610, which is on page 121 in your text. Again, you do not have to know, sorry y'all, my computer's being crazy. You don't have to know the 6 major steps, but I do like for you to see these, okay? So step 1, you can see right here, it's going to, bring that acetyl-CoA in, and then if we keep on going, steps 2 and 3, we're involving NADH, ATP, and CO2. They're all generated during the redox reactions, where the redox, we're taking on some of those hydrogen electrons. Steps 4 through 6 are going to further redox reactions, and that's when we end up with our FADH2 and some more NADH. So again, we kind of produce NADH twice during it, over here at the beginning and at the end. ATP comes out, CO2 is produced at the very beginning, and then FADH comes out at the end, okay? Now, step 3 is oxidative phosphorylation, and it says to define it. So it's stage 3. It produces most of the ATP in cell respiration. In the mitochondria, we're in that inner membrane. The electrons from NADH and FADH2, remember I told you those are like our little taxi cabs, they pull up to the top of the chain, they drop off their hydrogen electrons, and those electrons are passed down the electron transport chain, where they're picked up by oxygen to form water. Okay, so oxygen picks up the hydrogens, and that's how we form water. And if you'll notice, y'all, we haven't used oxygen at all until this point. So it's used at the very, very end of cell respiration. The energy that's released by these redox reactions is going to be used to pump hydrogens into the inner membrane space. This is going to start a process called chemiosmosis. Chemiosmosis is going to give us the most bang for our buck. So what happens, hydrogens are charged. So if you've got a bunch of hydrogens that have accumulated inside the space, you're going to have a gradient. You're going to have a hydrogen gradient, and that means you're going to have more hydrogens on one side than the other. Well, y'all know what gradients do. They make stuff happen. That's going to drive those hydrogens back through to the other side, but they go through this enzyme complex called ATP synthase. ATP synthase, remember it ends in ASE. We know that means it's an enzyme. This enzyme is going to become really, really excited, and when it becomes really excited, it makes a whole bunch of ATP, okay? I mean like 32 ATP, like a whole bunch. So chemiosmosis is really using a hydrogen gradient to energize an enzyme, which is ATP synthase, to make a whole bunch of ATP. So this shows you oxidative phosphorylation, and it includes 2 things, the electron transport chain and chemiosmosis. So this is on page 122 in your book, but again, we are in the inner membrane right here. We're on the inner membrane. Here's the outer mitochondrial membrane. We're on the inner mitochondrial membrane. That's where oxidative phosphorylation takes place. So NAD and FAD, that's what I call NADH and FADH2, the cousins, they pull up and they're going to drop off their hydrogen electrons. All right, well, those hydrogens are going to pass down the chain. They're going to go from this carrier protein to the next carrier, to the next carrier, on down until they're picked up by oxygen, right? Oxygen's the electron, it's going to pick it up, acceptor. It accepts those hydrogens, which is a redox, and they're going to form water, okay? Now, while that's happening, there are some hydrogens that are slipping through these little carrier proteins, and we've got on this inner membrane space, we now have a whole bunch of hydrogens, so we have a gradient. Well, they're going to energize this funky, looks like a turbine, sort of like it's a rotor, literally the rotor. It's going to energize this enzyme, this blue and purple thing. This is ATP synthase, all right? And when it gets energized, it is going to take all these hydrogens right here, and it's going to shoot them through this little enzyme, and that's going to take a whole bunch of P's and smack them up to a whole bunch of ADPs, and we're going to end up with a whole bunch of ATP, which is what we want at the end of cell respiration, okay? So excellent picture there. You can see it a little bigger on page 122 in your textbook, and it shows what's happening in oxidative phosphorylation. So the checkpoint question says, what effect would an absence of oxygen have on the process of oxidative phosphorylation? Well, without oxygen to pull the electrons down the chain, because in essence that's what it does, the energy stored in NAD and FAD could not be harnessed. So we would not have any ATP production. So basically it would be a bust. We wouldn't be able to finish the process without oxygen. Okay. Now, just as a review, the numbers. We're reviewing the numbers here. If you look at objective number 9, list the overall quantity of ATP made by cell respiration. That would be 32. And the efficiency of cell respiration, and that would be 34%. So with substrate level phosphorylation and oxidative, we produce up to 32 ATP molecules for every glucose oxidized in cell respiration. Okay, oxidize, remember, means to lose electrons. Reduction is to bring them on. So we get 32 at the end. When it's all said and done, we get 32 ATPs at the end of cell respiration. The efficiency of cell respiration is 34%. So 34% of that is converted to energy, and the rest, of course, is lost as heat. Okay. Now, your checkpoint question here, if you look at it, I was trying to tell you what, oh yes, we're on page 124. So the checkpoint says explain where oxygen is used and CO2 is produced in cell respiration. And so oxygen accepts the electrons at the end of the chain to make water. It's the official electron acceptor, whereas CO2 is released during the oxidation of those intermediate compounds during the citric acid cycle and when we're oxidizing pyruvate. So that's where those two things are used and made. Okay, so one more time, this is your numbers. So we get 2 ATP from glycolysis, 2 from citric acid, and about 28 from ETC and chemiosmosis. So that totals to give you maximum per glucose is 32 ATP. Okay? All right, we're going to stop there, and we'll finish up with fermentation